Okay, so we're going to start talking about the chemical logic for metabolism. So we've talked about this a little bit so far in kind of an introduction, and now we're going to start getting into the specifics, specifically talking about the process of cellular respiration. Coming up next, we're going to start with glycolysis, then talk about the TCA cycle, the electron transport chain. Essentially, we're looking at the processes by which we take the energy stored in the chemical bonds of our food and we transform it into a usable form of energy for our cells, which is mainly ATP, which is adenosine triphosphate. So we're not going to spend a lot of time on this lecture because a lot of what we're introducing in this lecture you're going to see specific examples of in the next lecture with glycolysis, the TCA cycle, and the electron transport chain. So to revisit what we had said at the beginning of the quarter, we said that the metabolism is the sum of all the chemical reactions in a cell. And this is just from the slides, but I'll write it because we'll kind of use it as a model. So this is complex metabolites up here, and then we'll say simple products down here. So when you're going from something that is complex to something that is simple, we're breaking something apart. Breaking apart. You could also call this degradation. We're taking something that's big and complicated and breaking it down into something that is small and not small and simple. So you could look up at this as like a polymer, and you're breaking it apart into a monomer, or even smaller, even into individual elements. When we're going from a simple product to a complex metabolite, we're building something bigger. This is called biosynthesis. And the words that we used to define these, and a process where we're breaking things apart, we said this is catabolic. And then when we're building something, this is anabolic. So when we are undergoing a catabolic process and we're going from something that's complicated to something that is simple, usually things that get made are ATP, and this is the major energy transduction molecule. This is kind of like the currency of the cell. So it is used to transfer energy. Another product of these catabolic pathways are NADH or NADPH, and we'll talk about those. NADH, NADPH. These are electron carriers, and we're going to see these, both of these, electron carriers. They carry electrons from oxidation reduction reactions to different parts of the cell. So the opposite is when we are building things, we are hydrolyzing ATP. So this would be ADP plus phosphate. 2 minus, sorry, and NAD plus or NADP plus. So these are oxidized, these are reduced, these are oxidized, these also electron carriers. So we're going to talk about a lot of these in the chemical reactions that we're talking about. So there are some really good slides in the PowerPoint showing examples of these processes. When we are talking about glycolysis, which is what we'll talk about first, so I'll use that as an example. Glycolysis is a catabolic process. It's catabolic because you're taking one glucose molecule, you're breaking it down in 10 steps. I'm just showing 2 arrows, but there's 10 steps. From 1 molecule of glucose to 2 molecules of something called pyruvate. So you're going from something that is more complicated to something that is more simple, something bigger to something smaller. During this process, we produce ATP. We also reduce electron carriers, and then we'll see that where those ATP and those reduced electron carriers go as we proceed throughout the rest of cellular respiration. Glycolysis is the process that we're going to talk about the most. Glycolysis is the way that we use carbohydrates or sugars for energy. We also will talk about how we extract energy from lipids and proteins, but we will not spend nearly as much time on those as we will carbohydrates and glycolysis. There's examples in the slides of different types of reactions to keep your eye on. Remember that these reactions are all dependent on free energy. Remember that living cells are not at equilibrium. Living cells exist at something called homeostasis. Homeostasis is not equilibrium. So that's something that we'll see. There's going to be some difference in free energy during these chemical reactions. And we will couple energy producing reactions with energy requiring reactions. There's a slide on slide number 10, which shows the structure of ATP. ATP is adenosine triphosphate. So it has 3 of these phosphate groups. The phosphate groups are connected to each other with a phosphoanhydride bond. And then there's a phosphoester bond between the first phosphate and the adenosine. When we hydrolyze ATP, so ATP hydrolysis, we have ATP plus water, and it's going to give us ADP plus inorganic phosphate. PI is how we represent it. This is a reaction that is going to release energy. And usually in like a biology textbook or other science books, you'll see some sort of like flash or like lightning bolt coming off when it shows this reaction, which makes you think that breaking this bond is releasing energy. But we know that breaking bonds don't release energy. Breaking bonds actually require energy. The reason that this over here has a lower free energy than this ATP by itself is because this phosphate, this orthophosphate, inorganic phosphate, has resonance structures. So the overall free energy of ADP and phosphate is lower than ATP together. So we'll talk a lot about ATP hydrolysis later when we get into glycolysis and see some specific examples. What else to say? Some things to pay attention to as we're going through a lot of these processes is to see when things are being oxidized or reduced. So oxidation and reduction is when we're transferring hydrogens, and there's a good example for NAD or NADP. This is on slide number 27, showing oxidized cofactors. So up here, these would be oxidized. And then the reduced cofactors are the ones that have the hydrogen. So having a hydrogen is a reduced cofactor. These are called cofactors, NAD and NADPH. We'll talk about those and see the structure of those when we get into glycolysis too. So things to pay attention to and to be looking for as we start going through these different metabolic pathways. One, you should be looking for any time there's a change in the structure of the intermediate. Intermediate just means anything that's not the initial substrate or the product. So back to glycolysis, glucose is the starting material, and then there's going to be 9 intermediates between glucose and the final end product of pyruvate. Glucose has 6 carbons. Pyruvate has 3 carbons. So at one point during this glucose to pyruvate, this 6-carbon molecule gets split and then eventually produces 2 identical 3-carbon molecules. So pay attention to steps like that where especially the carbon skeleton is changing. You also want to pay attention to any sort of phosphoryl group transfer. So for example, again, all of this is going to be applied in the next lecture when we talk about glycolysis, but the first step of glycolysis is glucose gets enzymatically changed to glucose 6-phosphate, where we add a phosphate group to the 6th carbon of glucose. Well, the phosphate group that we add to glucose comes from a phosphate, an inorganic phosphate that was cleaved off of ATP during ATP hydrolysis. So pay attention to phosphoryl group transfer, and that's something that I'll always bring up. Also pay attention to electron transfer with these electron carriers, so NAD plus or NADH. We won't do many examples with NADP or NADPH. Those are usually used more in anabolic processes, and most of the processes that we'll talk about in this quarter are going to be catabolic. Let's see, that is probably it for this chapter. We are going to apply a lot of this to glycolysis moving forward, so you'll see a lot of specific examples of this coming up.